This is episode 57 of the Immunology Podcast, Careers Away from the Bench with Drs. Laura Raff and Ashar-Patel. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where you have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Drs. Laura Raff and Ashar-Patel from STEMCELL Technologies here to talk about their pivot from the lab to sales. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
So we would like to remind our listeners about the upcoming International Union for Immunological Societies Congress taking place in Cape Town, South Africa, from November 27 to December 2nd. Jason and I will be attending. We just could not be more excited about this. So early bird registration until August 30th, and you can learn more at iuis2023.org.
Yep, one more big trip coming up.
Yeah. Oh, very exciting. But happy birthday, America.
Yeah,
July 4th.
Yeah, it is. We're gonna ignite lots of gunpowder in the air as we are opt to do and celebrate. So, it's the usual. Probably have a barbecue at home. Well, I'll just be getting back from a couple of vacations, so it'll be kind of chill, but we'll do something.
Very nice. How many years already? Like
1776, right? So
oh gosh, you're not gonna maybe do the math, right?
250 puts you at 2026. So 3 years shy of 250, so 247.
Okay, almost as old as your president. Sorry, what? No, I didn't say that. No, just kidding. But nice. So how much long do you think you guys have left? It's been a good run though.
Yeah, that's a good question. I don't know. Like we're having a little bit of geriatric disease. Maybe we just need we need the people at the other podcast to give us a stem cell infusion and get us back young again.
True. A middle-age crisis, but you'll get through it, guys. I know you will. We're all counting on you. So, yeah, did you know that Argentina's birthday is on July 9th?
Oh, really? No, I did.
Mm-hmm. That's our Independence Day. Quite close. So I'm also having some celebratory
how long ago
barbecue.
Was that?
That was in so it was in 1816.
So we're at 247 minus 30, 217. There we go. We were
following your example. I mean, actually, a lot of the way the Argentinian politics is organized is modeled after the American model. So,
interesting.
Thank you for that.
You're welcome. Sometimes you just got to go tell the British to take a hike and then things happen.
Yeah. We did ditch the whole, electoral, we actually directly vote for a president, but that's fine.
Yeah, we're weird. We're also really big, which is why they did it. People forget we're more like an analogy to the EU than not.
Yeah.
The state is kind of like a country.
Yeah. You like to think you're like, I guess it is a big country.
Technically it's true. They're all sovereign to a certain level.
Yeah, but it wasn't that big when it started, right? It was like just
13. So it expanded from, Maine all the way down to South Carolina and Georgia. So it's not that small.
All right. You know what's not that small either? Our selection of science for today.
Although we're going to try to be brief, that's true, but that was a wonderful segue. But I'll discuss some really small things first with the, cellular receptor level here. So, big concept, small signaling. Ooh. All right. I should stop before the dad joke police come and get me. All right. So this is it's in Cell. It's highly multiplex bioactivity screening reveals human and microbiome metabolome GCPRome interactions. First author is Han-Wei Chen. Last author is Noah Palm. Came out in Cell July 6th. So, which means this is the preprint or the early online first. This is really a technical paper. They use something called Presto Salsa. That is their technology. At a super high level, what they do is they take a GPCR, G protein coupled receptor. They have a library of them. And that has a second plasmid that has beta arrestin tied in the GPCR on the inside. They use the TEVs thing. So the tobacco enterovirus cleavage system that you use for protein expression, and then the cleavage site. And so what they have is they're on that cleavage site on the inside. So what they do is they have the TEVCS, and then this trans-activating region. So you have a GPCR with a cleavage site for the enzyme and a trans-activating region. That trans-activating region has the reporter in it. Okay. Then when you have GPCR signaling activation, you have, so you have the trans, then you have 3 plasmids. GPCR with a T8TA activator, transactivator. Second plasmid's the adapter, beta-arrestin 2 with enzyme attached. When you have G protein coupled receptor signaling, beta-arrestin moves over to the GPCR, and because the enzyme is there, it cleaves it and releases the transactivating region. Third plasmid has EGFP in it, activated by the transactivating region from the GPCR. It's cleaved, so it floats away and hits it with, in this case, a barcode. So now what you can do is you can put this into cell systems on a plate, 96 well plate, with each GPCR has its own reporter plasmid on it, and everyone has the same adapter. And you put a whole bunch of GPCRs and transduced on the different cells and hit them with the same agonist or the same bacterial lysate or whatever else. And you can figure out which GPCRs are activated, because you can then sequence everything. And because each one has its own barcode, you have an internal check for, does it glow? Yes, it worked. That well worked. But then you can pull it all together in sequence and use the barcodes to tell you if you have activation or not. And so now you can rapid screen GPCRs for things. And so in the way this works is they then went to the gut and bacterial metabolites. And this is more of a proof of concept, but they showed that various human metabolites could be, they could figure out that various human metabolites are being activated as you expect. They found that this specific microbiota bug, Porphyromonas gingivalis, which is an oral bug, has this protease gingipacan A that activates a specific enzyme. And this is kind of new, but made sense, or a specific GPCR. It kind of made sense based on other stuff. So they show, hey, we can find some new things, but this makes sense based on biology. But long and short is you can take the bacterial lysate of a bug or of a human gut, right? And then put that on there and say what GPCRs are activated. You can put specific bugs in. You can put beta agonists on there and see that those light up as controls. And so it recapitulates everything. Notice this is the GPCRs that are not the taste receptors or the smell receptors. So specifically, it's the non-olfactory GPCRs. Most of the nose is thousands, like all these GPCRs though. So this is everything else for good reason. So it's a really cool paper. It's a technology paper, but it's really neat. And I encourage people to look at it. Presto Salsa is what it's called. And then they found some microbiome links, which really was a sanity check, but showed you how fast you can do some stuff with it.
So how did they, what is that supposed to be an acronym for?
It is an acronym. So the original thing called Presto Tango. So Presto is parallel receptor ohm expression and screening via transcriptional output. That's Presto. Salsa,
in contrast to tango,
I can't figure out how they came up with salsa. It maybe is in the paper, but they don't have that nice and clean. Mm. It's built off of tango, so they called it salsa.
Yeah, because I guess I was thinking of salsa as like the sauce, but no, salsa is the dance style. Now I guess it makes sense. So just for me to understand, so this is designed in order, do you have a library and then you
have a library,
introduce one GPCR per cell, basically.
Right, per well, along with its Presto Salsa reporter plasmid and the adapter plasmid. So it's a triple transduction. And you get all 3 in there. Glowing tells you the system's working, but barcoding tells you what happened.
Okay. Cool. Okay, very interesting. Like, well, often you see this technology, new platforms that people device, and you cannot help thinking, man, how like this is very complex. Like, sometimes they have these really cool ideas, and it's really nice to see how these platforms come around. Although it must have been a lot of testing and benchmarking.
It definitely is, but like usually GPCR signaling has been stuck with radioligands, so this is a way around that.
Okay. All right. So I also have a paper that kind of is about technology in a way. But I also thought it was very interesting because it touches upon a subject that I really like and I think is extremely important, which is of course, well, vaccination in a way. So I always think that vaccinations probably one of the most important achievements of our of immunology and our study of immunology. And there's a particular one vaccination that was very has been very um what's the word, very iconic or very exemplary of what we can achieve. um Besides, well, for smallpox has been a huge success, but then I think another huge success has been poliomyelitis vaccination, so against polio, and how we have really swiped this disease from most of the world. Of course, there's still some pockets of wild type polio in a few countries, but it has been, when you hear the stories of those huge outbreaks in the 50s and the well and before, it was quite impressive what we have achieved. And so the thing with polio vaccination is that you might remember from your medical school training is that we have basically two ways that we can vaccinate against polio. And there were these two fundamentally different vaccination strategies developed. One by Jonas Salk, in which you have at what is called an inactivated polio vaccine, in which basically you take polio virus and you inactivate it with formaldehyde that prevents, kind of kills the virus in a way, prevents it from replicating, and just gives the epitopes for your immune system to be trained on. And that vaccine is inoculated through an injection. Um, and it's a very good vaccine in the sense that it gives you protection against, uh, the uh, the neural effects of polio, and the potential neurotropism of the virus, and then prevents paralysis and other issues with your nervous system. um, but it does not protect against kind of transmitting the virus. It's a virus that poliovirus really likes to home in your gut, and it will replicate in your gut, and that's why it also gets excreted from your feces, and that's a way by which polio is transmitted. Another vaccine that has been really kind of the workhorse of our efforts to eradicate polio is what are known as the Sabin vaccines that are made by a different mechanism, which was these strains that were identified by, what was his name? Sabin? I forgot his actual first name. Um, and he actually showed that he could find attenuated strains that would not generate poliomyelitis on the patients. So they would not, if you get this as an oral, you could take this virus in low doses orally, and it would replicate in your gut. It would generate a very strong immunity in the gut, and that would prevent you from, in the future, getting the polio virus and also letting it replicate in your own gut. And that had a very beneficial effect on reducing transmission of polio. But there's a huge issue with these vaccines. Although they are the cheapest and the most easy to administer, because you only need to like take up a couple of drops, and it has been used especially in kind of not developed countries to guide the efforts of vaccination. There is a huge issue, which is because the virus is kind of alive in a way, as alive as a virus can be. The virus replicates in your gut. Does not cause disease, but it does, because the virus, as any virus does, mutates as it reproduces. There is a chance that you will revert back to the original poliovirus that does have a neurotropism, that does affect your nervous system, and it will give you the disease of poliomyelitis. So a big issue has been that as less people get polio, these polio that derived from the vaccination becomes more and more important. And particularly in cases where you have a population that is vaccinated with this particular strain, and then there's people around them that are not vaccinated at all, these people are susceptible to this potentially reverted virus strain. And this has been the origin of a lot of small outbreaks that have happened throughout the years. And it is the source of some of the strains that have been seen in the last decades have been a consequence of reactivation from vaccines. So what can we do about this? Well, all the advancements that we have an understanding of how polio works, there has been a new vaccine that has been kind of designed more rationally to prevent this reversion to the wild type or reversion to a virulent strain. And this is kind of a new, it was known as a modified version of poliovirus. So in this case, you have 3 different strains, which have been the traditionally ones generating disease. We have mostly eradicated the wild type versions of strains 2 and 3, and strains 1 are still existing in small pockets in the Middle East. But then we have strain 2, which the Sabin version, the attenuated version, has the highest propensity to revert to a virulent strain. And recently, there has been a new vaccine available, which has been rationally designed to prevent this from happening. And this is basically what they did is they saw what are the modifications that the Sabin vaccine has that makes it not virulent as it is, and they found ways to lock this particular genome in place and prevented that individual mutations that happen as the virus multiplies to revert back to the virulent strain. So basically, you have nowadays it has been approved for emergency use an oral polio vaccine that is this new modified version they basically modify the genome in a way that particular one place called domain 5, which is very much associated with the virulence of the neurovirulence of the polio virus, and they have really locked this. And so far, there has been for a couple of years has already been used for vaccination when outbreaks happen, particularly outbreaks occurred by previous Sabin vaccinations, and has been very successful in both controlling the disease and then not generating viral vaccine derived infectious viruses. And so the paper here, following from the success of this type 2 polio's this vaccine derived from type 2 polio, they now designed the ones for type 1 and type 3. And this would allow us to vaccinate against a whole range of possible polioviruses that have existed, although type 2 and type 3 are not really found in the wild anymore. But well, you never know if something like that could come back. And basically what they show in this paper, and I know it's a very long introduction, because in the end, I think what I really liked is knowing that we are still trying with this. I mean, the polio vaccine has been out there for over 50 years. And how we can, by rethinking rationally these vaccines, we can bring up a new era in a way to solve a problem that was caused by the vaccination in a way, and by the success of the vaccination. So what they show in this paper is that similarly to what happened to the strain 2, the novel or oral polio vaccine derived from strain 2, they show that for strains 1 and 3, they can also generate vaccines that have a live attenuated polio virus that substantially reduces that has a much more replication. It has a similar replication fitness, so you can produce it in vivo cells similarly to the original Sabin strains. It has, when they tested in mice, they have mouse models in which they have introduced the polio receptor through the mouse cells, so they can use mice as a model for studying the virus and the vaccines. They show that these actually are more attenuated than these original Sabin vaccines, and that if you inject the mice with high doses of these vaccines, they don't, they're a lot safer to use. And then because they have much higher genetic stability, and this they do is particularly one of the things they do is they modify their RNA polymerase of the poliovirus to be more, uh, um, be more faithful in the replication, therefore reducing the chances of mutations. And they also prevent recombinations, and they change around the genome to reduce the chance of recombinations. And so they show that these vaccines as well are attenuated, and they have a much lower tendency to, uh, they don't revert back to the original, to really virulent strains. And even if they accumulate beneficial kind of mutations that give them a fitness increase when it comes to replication, they don't revert to all the markers of virulence that are important for the virus attacking the nervous system of a person. So I think the take home message from this paper is that they use mouse models, and they also show some data from the novel oral polio virus vaccine 2, that it has already been tested in Africa. And they show that by doing this rational design of the virus, you can actually improve substantially the safety profile of the vaccine, which has been a huge issue in our efforts to eradicate polio from the human population. Sorry, before I go to my second paper, I forgot to mention who were the talented scientists that carried out the study on poliovirus vaccines. So I just want to say a shout out to first author Ming Ye, and corresponding authors Raul Andino from the University of California, San Francisco, and Andrew McAdam from the National Institute for Biological Standards and Control in the UK. They were the authors of the paper that was published in Nature, and it was called Genetic Stabilization of Attenuated Oral Vaccines Against Poliovirus Types 1 and 3.
So it was just the RNA polymerase and a few other mutations. They do other stuff to make it not be able to be virulent again.
So basically, they have three main modifications that they do. On the one hand, there's a particular structure in the 5' untranslated region of the genome that generates a structure that is associated, that is required for the virus to replicate in neurons, and at the temperatures that neurons are in contrast to the temperature which is a bit lower, in which you find enterocytes and the cells in the gut. So by modifying this, and was also present in the original Sabin attenuated strain, and that's why the virus does not replicate so well in neurons, and it therefore is not so dangerous. And then there's a second thing that they do is a little bit more kind of dramatic. They change a complete recombination site that is present in the genome, and they move it to another part of the genome, and this prevents the virus from almost generating the correct virulent version with a single mutation. And this really makes it a lot harder for the virus to revert back to the original, to the virulent way. And then they also have these mutations on the polymerase that reduces the mutation rate, which of course is not good for a virus. It's against viral evolution, but it's good for us because we want that genome to be reproduced and nothing else. So those are basically the three main things that they do, strategies that they use to modify the genome.
Interesting. All right. Well, I don't even know if I have a segue I can use here from polio, but staying in the gut with gut infection, inflammation,
there you go.
Cytokines, I guess. I'm going to talk to you about a paper that's kind of made the rounds recently. It's in Science Translational Medicine. It's called Microbiome Alteration via Fecal Microbial Transplantation is Effective for Refractory Immune Checkpoint Inhibitor Colitis. So this is Taylor M. Hansey's the first author. Last author is Yinghong Wang. This is a clinical trial of FMT in people who received immune checkpoint inhibitor. therapies for cancer who then developed, after that therapy, immune checkpoint mediated colitis. And they showed that FMT in 12 they had a study of 12 patients, and 92% achieved IMC clinical remission. So 10 patients achieved it after the first FMT, 3 required a repeat, and 2 of them had no response. So they got 1 salvage out of the repeat for mediating. That alone is a big deal because it gets rid of one of the major side effects. But they also showed that people who were responders tended to have people who responded to the therapy, immune checkpoint therapy more robustly, tended to be the ones who responded to the FMT more robustly. So they tended to go together. And this is just a reminder, this is after failure of steroids or infliximab or Entyvio and vedolizumab for other treatment. They also then were able to link this to certain bacteria. So this effect is driven by increases on Concella and bifidobacterium, which were depleted in the responders before treatment. So that's another big thing that they were able to find. And this was also CD8 T cell dependent. So they were able to see a drop in CD8 T cells in the colon after FMT, but not in other populations. In particular, they couldn't find a signal. It's not a super long paper. They, as I said, found these microbiota that worked. There were signatures of it. But what's just fascinating is it was the single FMT was able to overcome this major side effect of it. And that the donor microbiome was very different than the microbiome of people undergoing this. And the donor microbiome had a different baseline characteristic than even people who are going to get the therapy, and then had a shift to, so 2 parts. One, if you were going to respond to the therapy, your microbiome was different than baseline to an extent. And if you got colitis, it was different, and it was very different than the donor they were getting. And so you're seeing a shift all the way back to a baseline, essentially, from this. It wasn't just like, oh, the drug screwed it up. The fact that you have this additional inflammation is also a sign that your microbiome was a little bit different from other people at baseline. But there was no difference in the diversity between patients with complete response or non-complete response or FMT donors at baseline. So, but the stool was distinct from the stools of people with complete response. But people who didn't have a complete response to therapy were not that different than the donors. So being someone who responded to therapy was a sign that you had a different microbiome to begin with. And then you also were different if you had the inflammation with the colitis, and then you could get back to normal afterwards.
And is it possible that this baseline microbiome of these different patients is associated with whatever
clinical response and stuff?
Yep. No, whatever previous therapies that they had?
Maybe. They don't get into that level of detail here, but they do think your microbiome at baseline influences your ability to have a response to checkpoint therapy, generally speaking.
Yeah, well, we did discuss that last episode, right?
Yeah, this is tying into the last episode, right? Where we know that, and now you're seeing a negative sequela, which is this gut inflammation, but then that also can respond to FMT.
And sorry, is this, which kind of cancers was it? Was just any?
Genitourinary, 50%, majority of stage 4 disease. So mostly it looks like genital urinary.
Okay. Yeah, I guess we go back to the point is that it does seem to matter, like, I guess, both for the efficacy of the therapy and also the kind of side effects that you get from it. What's in your gut?
It sculpts your immune system.
Oh, my.
We're just all little carriers. We're just mech brains in a meat suit carrying bugs around. That's our job.
Fair enough. Well, okay. So we're going to stay in the gut. Second paper is titled Dedicated Macrophages Organize and Maintain the Enteric Nervous System. Also published in Nature. First author Maria Francesca Viola from the group of geobukstangs at the Catholic University of Leuven in Belgium. So I think I'm going to try to be a little bit, give a high level idea of this paper, but I think very nice because we all know, I think we know by now that, macrophages, we always think of macrophages as this, bottomless pits of phagocytic cells that, eat bacteria and dead cells. And I think sometimes we do them a disservice by thinking of them as little trash, like walking trash cans. When actually they also have a very important role in pruning and in removing excess cells and excess things, and in particular, excess neurons or like synapses, and in this procedure they do is which is called pruning, like you prune an overgrown tree or something. They have a very important role in maintaining proper neuronal connections and removing excess. And this is now of the development in the brain, and and I think it's part of what we understand of how children's brain develops and things like that. But as I think you have said before, we do have a little second brain. Where is it, Jason?
In our guts. And by the way, macrophages are wonderful, wonderful cells and not just garbage cans.
I know. Sometimes I have to admit that I find myself underestimating them. So this is, a requiem for the macrophages. So we have this enteric nervous system, which is basically a collection of neurons, and is organized in two major hubs, two plexuses, myenteric and submucous plexuses. And in this case, they focus on understanding how this enteric nervous system develops in early age, and then they also discuss about another paper that they published in which they look at the importance of the organization of the ENS in later adulthood. But here we're going to focus on early. What they show in this paper is that there's a particular subset of macrophages, which are macrophages that are resident in the muscularis externa, so muscularis externa macrophages, and they are very important in organizing the ENS early in life. And they do this by pruning synapses and phagocyting neurons that are part of the system, and that the absence of their function has negative effects on, how the whole thing works. Like, for example, how well the movements of the bowel happen after in later life. So they do this basically by characterizing the ENS of mice at different stages. They focus on before weaning, so in the first couple of weeks of life, and they show that there are indeed these macrophages that are colonizing the gut before birth already. They already knew that they were required for the survival of these neurons from the ENS in adulthood. But when they look into how the ENS evolves in the early stages, they see that actually there is a reduction in the number of neurons and neural synapses between stage 10 and day 21. So there's kind of an excess, and then there's a reduction. And they show that this reduction is dependent on the action, or is associated to the presence and the phenotype of the particular phenotype of these macrophages that are resident and are associated to these neurons. They show that the absence of, so they can show that there's macrophages, phagocytized, which are phagocytical and are taking up neurons and from the ENS, and that they're important in order to ensure the correct development of these neurons there. And they show also that the development of these macrophages is in a way guided by the neurons themselves, by the ENS themselves, through production of TGF-beta. And that kind of this system regulates in a way. So I think that's basically what I wanted to say about this paper. There's of course a lot of details, very nice microscopy showing the direct kind of vicinity of these macrophages and the neurons. And they kind of make the point that we might be seeing something like, like a microglia situation in which the microglia prunes neurons in the brain. So you might be seeing something similar. And I guess it's not so surprising that it's a similar theme applies to our little second brain in the gut. So now macrophage is important to keeping your second brain properly developed and not overgrown in your early life, or at least in the mice.
Some of us on the stem cell immunology quiz are macrophages, and I would just like you to appreciate how important we are.
Of course you are. Of course.
And as my monthly reminder, enterocytes are just fancy macrophages.
Well, would I go that far? I don't know, but I'll give you this.
But if you're a TLR that intakes things, if a cell that has a bunch of TLRs on it intakes things and secretes cytokines based on what you eat, what are you if not just a fancy macrophage?
All right, I want to half accept that, just because I'm stubborn and I don't want to admit that you might be right.
It's okay.
And I'm a memory B cell, so we have
to remember things. You're going to hold on to that for a long time.
We move in different circles,
Fair enough. Well, speaking of different circles today, we're going to be talking about different career paths in science with Doctors Laura Raff and Immunology Ashar-Patel from STEMCELL Technologies. But before we get to that, have you ever considered moving from an academic career over into an industry position? Do you wonder what would be the right fit for you or not? Many stem cell employees start in academia and successfully transition to fulfilling careers in biotechnology, either inside or outside a lab. Listen to some of them describe their personal journeys by visiting www.stemcell.com/academia-2-industry.
Hi, everyone. We are joined today by two special guests. We have Dr. Laura Raff and Dr. Ami Ashar-Patel. They are both employees at STEMCELL Technologies, and they're going to talk to us about their experience working in the sales departments at STEMCELL. Laura is Associate Director of Sales of the Immunology area at STEMCELL. She has a PhD from the University of Southern California. And Ami is Senior Account Manager also at the Immunology area of STEMCELL. And she has a PhD from the University of Massachusetts Medical School. And we're very happy to talk to you today. And we're looking forward to maybe hear a little bit about your research arch, or how did you move from academia to industry? What do you like about your position? So welcome to the Immunology Podcast.
Thank you. Happy to be here.
All right. So to jump in, maybe we can take it one at a time, but Ami, could you talk first? What do you do now? I think we'll start with what you each do now and explain that, and then we can talk about how you get there, because I think knowing the end may be better in this case first.
Yeah. So as Brenda mentioned, Senior Account Manager is a fancy way of saying that I work with scientists in the biotech area. And I work specifically with any scientist in the immuno-oncology field, CAR-T therapies basically. And I talk to them about their research and figure out if there are any products from stem cell portfolio that they could be using in their research. So that is one aspect of it. The other aspect is, of course, troubleshooting. We already have customers who currently use our products. So if they run into any issues or if they are unsure of how a particular reagent works, I'm there to help them. I go into the lab sometimes to demo some of our reagents. I can talk over the phone with them, guide them through the protocols. And essentially, there's a lot of communication that happens over emails as well. Does that help? The short and sweet of it.
I think so. And how about you, Laura?
Yeah, so I am lucky enough to actually work with Ami along with 5 other sales representatives in the region as an associate director. So I really help support Ami and her colleagues supporting the scientists. So I join Ami on site visits. I help with more complicated demonstrations of how to use the products, help with some troubleshooting. So I do still work in the lab and interact with the scientists occasionally. But another big aspect of my job is also just supporting the account managers and helping them strategize and think more in the business sense as well.
Maybe I would like to ask, is there any particular reason, any particular circumstances that motivated you to make the jump from your academic background, from your PhDs, and also from any career that you had after, and that you think maybe an experience that you think might be useful for our listeners to hear about your transition story?
Sure. Yeah, I guess I can go first. As you said, I did the PhD and postdoc, and after that, I actually went into industry, but as a scientist, and worked at a company out in Santa Monica developing antibodies for oncology therapeutics. And I loved that. I actually loved being a scientist, leading different programs, and also doing the wet work at the same time. But decided to explore other opportunities and ways to support and be involved in science without being on the bench when my husband matched for residency. And we were forced to make a move across the country. And so I did a lot of informational interviews to just try to learn about alternative careers that still, allowed me to utilize my PhD, all the training I had, but outside and off the bench, I guess, at the end of the day. And I came across actually a scientific sales representative position at STEMCELL. I'd used STEMCELL's products before and thought, this is an interesting opportunity. I can absolutely apply the immunology and all the research I had done up until that point in a different way and really understand the business side, which was appealing to me. And so I applied and actually just fell in love with the culture of STEMCELL, with the motto and the drive of STEMCELL, which is scientists helping scientists, and took a leap of faith, I guess, at the end of the day. I went with my gut. It was not an easy decision by any means, but at the time I thought, okay, I can do anything for a year, and if I hate it, I can go back, which everyone says you can't go back. But in all honesty, yes, you can. You can absolutely go back if you really wanted to and make a career move. But I loved it. It was amazing to get into many different labs, not just focused on one small project that you're hyper focused on. I love learning about all the different groundbreaking research that's happening both in academia, but also in biotech and at pharma. And I've stuck with it, and I've been at STEMCELL for 10 years now in the sales department. Yeah. Ami, do you want to share, I guess, how sort of your transition is a little different?
Yeah, sure. So I'll answer this question in two parts. So I don't know, a lot of people don't know that I actually worked in industry before restarting my second attempt at PhD. So I was working as a research associate in between degrees, and I knew at that point that I always wanted to have like a more translational research project, and I knew that I would be better suited in a biotech or a pharma company. So there were some difficulties I ran into in terms of just the market that was at the time when I left pharma. And then I actually came back to academia to restart my PhD. And initially, I knew that I would want to go as a scientist back into pharma, but as it happens many times with people over the course of my PhD and postdoc, I realized that maybe I don't want to go back to being a bench scientist. I just knew I wanted to be in biotech or pharma. And similar to Laura, I did a lot of informational interviews during the last couple of years of my PhD and even during my postdoc to figure out what kind of role or career would be better suited for me. And I had a checklist, so sales was never there in my radar, to be honest. I had never thought of a career in sales. So I did not have many informational interviews with people in sales, but I did chat with a lot of people and kind of figured out like there are some careers that are absolutely not going to work for me. And at the same time, I was also attending career fairs, and I happened to meet a couple of, actually stopped by at the stem cell booth, and I met a couple of really great people, Ting Ting, who is not here anymore, and Lynn, who is also currently at Stem Cell. And we chatted and they talked about their roles, and I was almost sold. I was like, all right, this sounds like perfect. There's people and there's science, and those are both aspects that I really enjoy. So I, again, like Laura, took a leap of faith. I was like, let's apply, let's see where this goes. And I gave myself a year. I was like, okay, so let's see in a year if I still enjoy it or not. And it's been 5 years. I'm still here, loving it. No regrets.
No regrets. I like that. Jason feels, he identifies with the feeling, don't you?
I jumped from academia to industry when my PI went away one day to another country and didn't come back.
He was ghosted by his PI.
Yeah.
Sad. Very sad.
But you talk about troubleshooting and that being a big part of your job, which sounds like there's some science involved there. I wonder if maybe you could talk about that a little bit, because I think a lot of listeners have used stem cell products or other products from anyone and had to call up tech support sometime. But I think most people know it from one end. How does it work on the other end, especially when you're like, it's a customer service, so you can't just tell them to pipette it the right way if they're screwing that up, right? Like, or what have you. There's obviously some sales and people skill, but also there's probably some like things that you guys don't see, or you find problems and other stuff. So I don't know if you could deep dive on that a little bit. I think there's a lot there.
Sure, I guess I can take this one first on me, and then you can jump in if you feel differently. You're right. We can't ever go in and just say, well, it's the end user, and say that's what's happening. But oftentimes with troubleshooting, I just put on my scientific hat again as a scientist, and usually there is, some small tip or trick that we, have discovered over the years, utilizing the stem cell products that we can bring to them and share with them, to help sort of turn the experiment around. It usually is something really small that people don't realize is a big deal, or they, have veered away maybe from the protocol slightly from what was stated, and made a few changes. And if you just help redirect, that usually does work. But also a lot of the troubleshooting that we do is not the basic. Like, a lot of people are obviously doing things a little bit off label, almost like isolating a cell type that we don't have a kit for, and we're just helping them optimize. So it's putting two different brains together, being creative, thinking of different ways of doing it. And so it's usually it's not always just the end user, right? It's more just being creative as scientists together. And I think if you approach it from that sense, it usually works out pretty well in the end. I don't know, Ami, what do you think?
Yeah, it's pretty similar, I guess, thoughts. Of course, we can never tell the customer that they're doing something wrong. And also they have experience, they have used different products. So, and they are scientists after all. So I'm sure everybody has a way of doing their experiments. But sometimes it does happen that a customer is probably misreading something in the protocol, or if they are so used to doing certain things their way, they might not realize that some of the steps are different in the protocol, or they may have missed something. So basically, when we offer troubleshooting, we just end up going into the lab and doing the assay, like experiment with them, or we ask them to do it, and then we provide suggestions and tips. And that oftentimes helps a lot. And customers do appreciate us coming in the lab and doing like hands-on experiments with them.
I guess it's hard to resist the impulse of saying, sir, have you actually read the insert in the kit? It's like, the good old, did you turn it on and off to fix this? But I guess that oftentimes when you are a consumer, on the other end, sometimes I feel a little bit, I don't want to say I'm not going to call them for this. Like, I'm sure it's me. Like, sometimes I feel a bit hesitant to contact the customer service, or I always think maybe it's not, this is a small thing, I'm sure. So what would you have? Would you have any advice for those people that are struggling? Like, how do you feel? How do you like when customers reach out with, valid or kind of genuine questions, or how does it feel from your side, being there and troubleshooting with a customer, with a scientist that has an actual genuine issue? How is it from your side, the experience?
I can take this one. I will say I love it. I would rather have the customer reach out to me directly and ask me something rather than self-doubt. To be honest, I think that's the best answer. I love it. I have a lot of customers who directly reach out, and they may say, like, I'm not sure what's going on. It could be me. It could be the product. I would rather be there to help them and eliminate some of the things, and really showcase the product and make sure that they are, in the end, getting the results that they want from our products. So I will say, please do not hesitate. Reach out.
Yeah, I'll just add that it's like, why reinvent the wheel? These kits are and products that STEMCELL offers are of like the highest quality, and we've tested them internally so much they should work really at the end of the day. So there are so many other challenges as a scientist that you face each and every day in the lab. So yeah, absolutely reach out, and we are more than happy to assist for sure.
So I don't know if Jason has a question. Otherwise, I'm going to just quickly ask on that, kind of related to that. What do you think is the biggest misconception about your job that people that don't do it have?
Yeah, I can answer this as well. I think initially when I told people that I was moving away from being a scientist and going into sales, there were a lot of raised eyebrows, like within family, friends, even like professionally. And I personally think like it's not valid. I will say, like, at the end of the day, you can contribute to science in many different ways, and I think it's important to have an open mind. At the end of the day, I am still a scientist, and yes, I am doing sales, but that doesn't mean that I'm going to be a salesperson before a scientist. I don't know if that helps, but I think that's something that people need to just self-realize, that there's a reason we are scientists on the other side of the bench this time around. Like, we are there to help other scientists make sure we know how it was on the other side of the bench. So we are here to help, and we are not always selling. I feel like a lot of people think that, oh, if I contact my salesperson, they'll like ask me to buy something, or they'll talk about some other product. So at least at STEMCELL, I would say we are not like your typical salespeople.
Yeah, I would say it's not the dark side, but that's something I got a lot. Oh, you're going to the dark side, and at STEMCELL, it really isn't. Alan Eves, who started the company nearly 30 years ago, he's a MD PhD himself. He's a scientist, and really his whole mission is to provide like the highest quality products for people to help just make their research easier. And we are scientists there, yes, selling the products, but really we're there to help support the research at the end of the day. So as Ami said, it really is more based on the science rather than like just making money. It's really not that at all, which I think is more the misconception. We're there to really support the science.
So along those lines, and I don't know how much you can disclose, have you ever had, is there a story you can share of like a customer question that made STEMCELL change something that was really cool? Like the customer was doing something and it wasn't working and you fixed it and now it's a new product or something like that you can share? I don't know if you can or can't because sometimes it's proprietary, but any of those like cool, like back feed from the customer perspective that you guys obviously would be involved in?
Yeah. So we have actually many examples. We receive customer feedback all the time and take it into consideration for product development. That happens on a daily basis. We even have a feedback mechanism internally from all of the sales representatives in the field working with the customers to bring that back to R&D to help drive that product development. So that is ongoing. It happens every single day. We also there are licensing opportunities as well, where something is being developed in an academic lab or another lab. That's, wow, that is so cool. And it could benefit so many more people. And so being able to turn that around and be able to really support the greater scientific community through that licensing opportunity. I'm going to refrain from giving specifics because, again, I'm not 100% what I can, 100% of what I can and cannot share, but absolutely, we get the vast majority of our product development ideas from the customers themselves.
So what do you think are the best parts of your job? Like, so interacting with customers, but maybe could be something more mundane or it doesn't necessarily have to do with the job itself, but about working that is different to the academic style. I think people often mention things like, contracts and work-life balance. How do you feel about that from your side? How much of that do you think is true, or is really something that you notice as well when you did the transition?
Yeah, I will say the flexibility and like owning your own schedule is a big thing for me, I think. In academia, I know we have a flexible schedule. Like some people like to come later in the day, some people like to have an early start. But I think in regards to like the sales job, as such, it's flexible in the sense that I can meet my customers at whatever time works for me. So there is no I will say like expected time that I should be meeting a customer at whatever, first thing in the morning or like later in the evening. Like I can reach out to the customer and we can both figure out a time or a day that's flexible and works for both of us. So I enjoy that. And the second aspect, the work-life balance really is there. Like I really like having a Monday to Friday job. There is no expectations from STEMCELL that you're supposed to reply to a customer in the middle of the night or like, I don't know, reply to them over the weekend, for example. And those were some of the things that I experienced in academic setting where I was expected to answer my PI's email or like you just felt the pressure that you are supposed to respond to something that your PI asks you, even if it's the middle of the night, for example.
Yeah, I love the flexibility and the work-life balance as well, because I found that to be quite difficult as a junior scientist, I would say. And the work-life balance at STEMCELL and within sales has been wonderful, especially as a parent. It is helpful to be able to do both jobs, I guess, at the end of the day. But for me, it's the relationships. I absolutely love all the relationships I've built within the scientific community and within internally within STEMCELL as well. That's my favorite part of the job for sure.
Right. Well, we're going to have to wrap up. But before we do, we'd like to ask one quick question. And in this case, I'm going to ask each of you, if you could have one superpower, what would it be and why? And I know I'm putting you on the spot, so pick any one of them, any superpower ever.
Reading other people's mind. That's what I love to do.
Are you sure about that? Maybe, but maybe not
At
all
least
the time.
I know. Yes, is this person convinced by what I'm saying? Are they really going to buy? Are they really going to place the PO? Like, it's just good to have that clarity up front. Like, I would love that.
Yeah, within sales, I guess, I mean, that makes the most amount of sense, right? Yeah, being able to read someone's mind for sure. I would also love to fly, just to feel I like to be on the water, on a boat, feeling the wind. So I'm going to go with, I would like to fly.
All right. Yeah. Well, there we go. Well, thank you both for coming on. It's a pleasure.
It was great talking to you.
Thank you so much.
Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com if you have feedback or you would like to suggest a guest. See you next time.